Yeah, I want to return to the subject of buffers and acids and how they work in acids and bases, and be sure that you do have a thorough understanding of how buffers work. So again, reviewing very quickly then, the role of a buffer then is to maintain the pH of a solution. So again, that's not necessarily to keep a pH of a solution at neutral pH, but keep a particular solution at a particular pH or within a particular range of pH. And different buffers will work within different pH ranges. And then another thing I want you to keep in mind as we look at this example or these examples is that it's the hydrogen ion concentration of a solution that determines the pH of a solution. And so we'll basically be counting hydrogen ions and using that as essentially a measure of pH. But it's the concentration of hydrogen ions that determines the pH of a solution. So if the hydrogen ion number or concentration changes, that means the pH of a solution is changing. And then again, most of the buffers in the example that we're going to use are weak acids. And what that means then is that a strong acid will mean that it completely dissociates in a solution. So all of the acid will dissociate to release hydrogen ions. In a weak acid, only some of the acid molecules will dissociate to release hydrogen ions. So again, we'll see that in the example that we use, which is carbonic acid. So again, we're going to be looking at an example that actually works in our blood to help keep our blood at a particular pH, and we're going to be looking at carbonic acid. And so carbonic acid in solution then will at least partially dissociate to form hydrogen ions and bicarbonate ions, HCO3. And so again, I want to have you look at this kind of as a visually rather than worrying about the formula so much. And so this will represent H2CO3. So in these diagrams then, carbon is going to be black, the hydrogens are white, and the oxygens are red. So this would be H2CO3. So that's the carbonic acid, intact carbonic acid. The bicarbonate ion is going to be HCO3, so HCO3. So that's the bicarbonate ion. Hydrogen ions will just be the white spheres. I'll also be talking about hydroxides, and so that will be an O and an H. And then also we'll show a few water molecules, or I'll show you a water molecule too, so that would be H2O. So again, this is kind of the key for what the diagrams and animation, not really an animation, but at least the PowerPoint that we're going to be using here, and which is also in your lecture outline. Okay, so again, looking at it visually then, the carbonic acid then can dissociate to release free hydrogen ions and bicarbonate ions, and can also reform to regenerate carbonic acid. So the reaction can go in either direction. And what influences that basically is how much hydrogen ions or bicarbonate ions actually are in the solution. So if you were to add materials on this side, it will drive it this way. If you were to remove materials from this side, it will drive it that way. So again, in this reversible reaction then, again, what can happen is that the carbonic acid can dissociate to release hydrogen ions, forming bicarbonate. And then under other conditions, the hydrogen ion will reform with the bicarbonate to form the carbonic acid. So again, that's what's meant by a reversible reaction. And in some conditions it will go one way, and in some conditions it will go the other way. All right. Now let's go ahead then and look at a solution, make-believe solution if you will, and what would happen if we added a base here. So again, if we look at this solution, this would be a tiny portion of a much bigger solution, of course. We do have carbonic acids, intact carbonic acids, we have a couple of bicarbonate ions, and we have a couple of hydrogen ions. So again, this solution will have a particular pH, and it doesn't really matter what that pH is, but what's important about that pH is that in this case we're looking at 2 hydrogen ions in solution. So again, the role of a buffer is to maintain the pH of a solution. So at the end of this process, we have, we're starting with 2 hydrogen ions, we want to end up with 2 hydrogen ions. So if you're adding a base to a solution then, you're essentially adding hydroxide ions. So if we add a hydroxide ion to this solution, what would be the reaction of the buffer? Well, hydroxide ions are very reactive. They tend to react with with hydrogen ions rather. So the hydroxide ion, the OH, will react with a hydrogen ion, and that will form water. Now notice when that happens, what's happened to one of our hydrogen ions? It's no longer in solution. It's now been incorporated into a water molecule. So whereas we started with 2 hydrogen ions, right now there's only 1 hydrogen ion. So that means the pH has changed. So the role of the buffer is to maintain the pH, so it's going to have to somehow replace that hydrogen ion. So the next step here then is that one of the carbonic acids then will dissociate to release another hydrogen ion. And so now then we get back to having 2 hydrogen ions. So again, this would be a solution filled with lots of water molecules. I can't really show you all those, but that's basically the process. So I'm going to go a little more quickly through it this time, looking at the same situation. So let's go back to our original situation. Let's add a base. We've got our 2 hydrogen ions. So here comes the hydroxide. It's going to bond with one of the hydrogen ions to form water, and in response, one of the carbonic acid will release a hydrogen ion, and we get back to the original condition. Okay, conversely then, what if we add an acid? So again, we're looking at the same kind of starting situation with our carbonic acids, bicarbonates, hydrogen ions. We've got 2 hydrogen ions. And so what if an acid was introduced to this solution, and an acid then would bring in hydrogen ions? So we started with 2 hydrogen ions. We've now got 3. That's a change in the pH. We've increased the acidity here. So how can the buffer compensate for that? Well, bringing in hydrogen ions will cause hydrogen ions to bind to bicarbonate, reforming carbonic acid. It gets us back to our original condition, maintaining the pH. And again, looking at that one more time. So again, if we add the acid, we're going to be bringing in a hydrogen ion. The increase in hydrogen ions will force some of them into the bicarbonates, reforming carbonic acid. So anyway, hopefully now you have a better idea of how carbonic acid helps maintain pH in our blood, even if bases are added to our bloodstream or if acids are introduced into our bloodstream.